Alright, so we just finished this nature versus nurture debate. You could go on for days and days about this. Just to give you a couple examples here, a trait of something that's based only on genetics. An example would be something like blood type. If you're AB or O negative, there's nothing you can do environmentally to change that. In other words, once you're born with that, nothing you eat or drink or do anything at all could ever possibly change that. That is based solely on your nature, nothing to do with the way you're raised. Something that is based solely on the way you're brought up would be things, I mean, it gets hard. You can't even say things like political views because, yes, a lot of it's based on how you're raised, but, I mean, there are people who just naturally see a certain way as far as the way people should be treated and how the government should spend stuff. Something that's based solely on nurture would be the language you speak. It doesn't matter what your genes are. Your language is based solely on where you're raised and the people who are around you. Plain and simple. Nothing to do with genetics at all. So let's move on. Gene interaction. You can have basically the way different genes interact. Like, here's a good example. You might go back to our skin pigment example and have 4 dark melanin genes. Now, remember that with the skin deal, with the pigmentation, the stuff we were talking about back here, this is not a matter of dominance and recessive. It's not like, oh, this is a pale person, this is a dark person. You just basically have 6 possibilities, and every time you have a dark pigment gene, you throw a little dash of melanin in, little dash, and your mom gets 3 dashes, and your dad gets 3 dashes, and maybe your mom has 1, 2, your dad has 1, you got 3 to 6, you're somewhere in the middle. Maybe dad donates all 3 and mom donates all 3 and you're super black. Maybe mom donates none and dad donates none, you're super pale. It's just how many of those did you get? However, if you have a person who is maybe of African descent or someone who grew up in the Dominican Republic and it's like dad donated all 3 dark pigments and mom donated all 3 dark pigments and his baby's going to be super black, oh, but they're albino. It doesn't matter. There are cases of people who are African but are albino. And it's like weird. It's a white-black guy. That does happen. And that's gene interaction. In other words, it doesn't matter what your pigmentation genes are. This other gene, this albinoism, steps in and fixes it, or masks it, really. Environmental effects. Here's a good one. Sometimes genes will not be fully expressed owing to some external factor, like human height. Your mom might be tall, your dad might be tall, but if you're raised in an environment where you are malnourished, you will not grow to that full potential. And you'll be like, oh, that's weird. Tall equals short. No, genetically they're tall. Environmentally, something's prevented that from happening. So now let's get into the really crazy stuff here. Gregor Mendel was kind of excited to combine his purple flowers with his white flowers, thinking, we talked about it, you might get something tie-dyed or striped or spotted. You might get some flowers that are purple and some that are white. You might get lavender. You might get some that are purple, white, and lavender. You might get some that are purple with lavender and white spots. All these things can happen. Well, when you're dealing with this thing called incomplete dominance, it's kind of unique. In this case, what's happening is you take this plant that's red and white. Red is not fully dominant over white. It's not completely dominant, and we say it's incomplete dominant. Or red is only partially dominant. Or as I like to say, incomplete is in between. Do not get this mixed up with the next one we're going to talk about. Incomplete is in between. A red and a white will basically cross and make something pink, which is in between. So red is not fully dominant over white. Now, you have to express this in a completely different way. Here's your P1, F1, and F2 generations, okay? We're going to use C for color, and then you still get 1 trait from mom, and 1 trait from mom, and 1 trait from dad. And this is a red from mom, a red from dad. Over here, a white from mom, a white from dad. And when you blend them together, there is no real hybrid. The hybrid would be, oh, we got 1 red, 1 white, and it's color red, color white. So if you see these little kind of exponent looking deals, these, I don't know, there's no other way to really say it. you know that you're dealing with incomplete dominance. And if it's, the hybrid essentially ends up being this pink, this in between. Well, then if the F2 generation, if you were to combine these pink, and you can look on the outside here, this ova, the female, this is a red and a white, so a pink female with a red and white, it'd make a pink male, pink female, pink male. Well, you'd have this 1 to 2 to 1 ratio still. 25% would be red, 25% would be white, and 50% would be this kind of hybrid. And this is kind of cool because now there are technically 3 phenotypes. You have the red phenotype, the white phenotype, and the pink phenotype as well. So it's something really complicated. Think how much this would have thrown Gregor Mendel off. He'd been like, oh, there's 3 genes. There's a red gene, a white gene, and a pink gene. There's not. You'll notice up here that we did not have a CP for pink. Pink is not a real gene. Pink is this illusion of a gene because it's what happens when red and white mix. It's when you have one red and one white trait. It's a red allele. That's a better way to say it. There is no pink allele. When we define the word allele, it's like an option for a trait. There is no pink allele. And that would have really bugged Gregor Mendel. This pink is just a combination of having one red allele and one white allele. So there's still just 2 alleles, but there are 3 phenotypes. Now, if that didn't confuse you enough, let's talk about something that's co-dominant. If you ever watched The Office, you know that at one point Jim Halpert and Michael Scott were co-managers. What does it mean to be a co-manager? Who's the real manager, Jim Halpert or Michael Scott? Well, they're co-managers, which means they're both managers. There's a concept called not incomplete dominance, but co-dominance, where red and white are co-dominant. Remember, the dominant gene is simply the one that's expressed. Purple and white. Purple is dominant, only purple shows up. It's dominant. So if red and white were co-dominant, what do you think would happen? Which one is dominant? They're co-dominant. They're both dominant, and both would show up. This is co-dominance. And the way they express this makes a lot of sense. You use 2 different letters because you always have the capital letter that is dominant. Well, red is dominant, and white is also dominant. So these hybrids, this red and white, the hybrid in this case, both show up. It's not something in between. There are sections that are clearly red, and there are sections that are clearly white. Red and white in this case are co-dominant. So if you see these little subscripts, these little exponents up top here, you'll know you're dealing with incomplete dominance, and incomplete is in between. And if you see where you have multiple capital letters, you'll know you're dealing with co-dominance, and both are dominant. In this case, red and white make red and white. So both letters will be capitalized. Keep that in mind for the worksheet you're going to do a little bit later. Lastly, let's take in one more concept. So we've talked about cases where you can have multiple genes, polygenetic, many genes controlling one trait. You can talk about the environmental impact of this. So, if you plant in certain kind of soils or spend more time in the sun, genes are going to be expressed differently. You can talk, that's how the whole nature versus nurture debate. You can talk about incomplete being in between. You can talk about codominance where both of them show up. The last thing we're going to show up is this multiple allele. This is where, in the case over here where there's a red and a white, there's not really a pink. Well, there are actually alleles where you have many options. You have a red, a white, a pink, a blue, a yellow. You have lots of options. Remember, alleles are options. Take the human blood types, for example. There's A, there's B, there's O, and there's AB. How much time do I have? All right. To understand this, there's kind of a weird concept to bring up, and it's that cells on your body have little proteins that tell your body itself. It's basically an ID card that says, I belong here. Okay? Those are basically antigens. They're just little markers that stick on the outside of things that say, hey, I belong here. I belong to Mr. Arkwell, or I belong to this student, or I belong to that student. My antigens are going to be different from your antigens, okay? antigens are also on the outside of foreign objects that say, I don't belong here. Now, they're not, a bacteria doesn't go into your body with a marker that says, hey, I'm foreign. It's just that your body's constantly looking for things, going, I don't recognize those markers. So why do bacteria have them in the first place? So they know each other, like, hey, he belongs, he's one of us, because essentially most bacteria are clones of themselves. So it's a way for it. It's kind of like in war. Why would you want to wear blue and the other team wears red? Well, so that your own team doesn't shoot at you. So it's basically a way of identifying what side you're on. And if you make antigens, you also make these things called antibodies. Antibodies will match up with antigens, and it's what we use to fight these off. Antibodies are made by your body, and they destroy foreign non-self things. So, like, if this is my antigen marker, these antibodies right here would not be in my body. But if this right here is an antigen from someone else, my body will make these antibodies to destroy this. You would not want antibodies to destroy your own cells. Those are called autoimmune diseases, and they're not fun. So that's basically the antigen antibody. You can see there are tons of different antigen possibilities. They're all based on shape, and they're very specific. One antibody will only bind to one type of antigen. So when you get sick and you encounter a new antigen, your body spends a lot of time trying to find what antibody will match up and destroy this. Kind of like a lock and key kind of thing. How about I move this right here? So this is the antibody, which is going to be our targeted weapon against this. Okay, why are we talking about this in genetics? Just wait a second. So here's this bacteria, and here's this cell that shoots these antibodies, and you can see the antibodies bind on via shape, and they either start killing the bacteria when they bind, they poke holes in it, or sometimes they act like homing signals to send white blood cells in to attack and destroy this. So there's the antigen, there's your antibodies. Remember, some antigens are going to be on your cells to say, don't shoot, I belong here. And you would never make antibodies to destroy your own cells. Here's a whole bunch of cells with these antigens, and here's the antibodies attacking them. They form clusters and really kind of bind things up. If you've ever been sick and the doctor feels you right here, they're feeling for clusters of antibody-antigen complexes, and it looks like this. So what sucks is if you have someone who has B blood and you give them A blood, well, you're not going to recognize that antigen, and your body is going to basically attack those cells as foreign. I'm used to seeing A blood. You suddenly put B blood in. I don't recognize that antigen. I attack and destroy it. So you have to, if someone has A blood, you can only give them A blood because A doesn't look foreign to them. If I have A blood, I don't produce A antibodies. Let me move this out of the way here. If I have A blood, I don't produce A antibodies. I produce B antibodies. And if I have A blood and you put B blood in me, I'm going to produce B antibodies and destroy that. And you're going to have these huge clusters forming, clogging up everything in your body. No good. So I have B blood. Someone puts A blood in. I start to have a big time immune response. So what this looks like, if I can do this in a minute and a half, just imagine this is A blood. Person that has A blood has these little antigens sticking out here. That's what looks normal to them. A person that has B blood has these completely different types of antibodies on them. This is what you normally have. This looks different. Well, if I put this, B blood, and someone who has A blood, I'm obviously going to attack them because I've never seen this before. And if I have B blood and you put A blood in my body, well, this looks foreign to me. What do you think AB blood is? AB blood is when you have both. You have these and those. So if you have AB blood and you give someone B, that's totally fine. I've seen these before. And if you have AB blood and you give someone A blood, that's totally fine. This doesn't appear foreign. But if I have A blood and you give me AB, this looks foreign. This is okay, but I'll still attack you. You want to guess what O is? O is when you don't have any of these. Everything looks foreign to you. Okay? Now, the nice thing about O blood is I can put O blood in AB, B, and A. So O is known as the universal donor, whereas AB is known as the universal recipient because that doesn't look foreign, that doesn't look foreign, even this doesn't look foreign. So if you're in a car crash, you hope you have AB blood because any blood they put in you is okay. And blood banks always want O blood because it's the universal donor. I want you to take a few minutes and follow along these genotypes here and look at these as well.